I was busy thinking about boys, boys, boys. I was busy dreaming about boys, boys, boys. Head is spinning thinking about I need that bad boy to do me right on a Friday, and I need that go.
Photosystem is a reaction center complex associated with light harvesting complex. The function of photosystem is the first one to trap light energy and transfer to spatial chlorophyll A. Second, absorb excessive light that damage chlorophyll.
In plants, photosystem is located at the thylakoid membrane, and this is how photosystem looks like. First, we will look at photon. Second, this is light harvesting complex. And third, this is primary electron acceptor. And next, this is reaction center. And not forget about the pigment molecules. And lastly, this is spatial chlorophyll A molecules. There are two types of photosystem in thylakoid membrane, which is photosystem 2 and photosystem 1. For the photosystem 2, the reaction center chlorophyll A is known as P680, while for the photosystem 1 is known as P700. These two photosystems work together to use light energy to generate ATP and NADPH.
I will explain about the differences between non-cyclic electron flow and cyclic electron flow. For photosystem, non-cyclic involve photosystem 2 and photosystem 1, while cyclic involve only photosystem 1. Photolysis of water occur in non-cyclic but does not occur in cyclic. Non-cyclic release oxygen and cyclic does not release oxygen. The electron flow for non-cyclic is in linear motion, which is start from PS2 and end at NADP reductase. For cyclic, it is in cyclic motion, which is start from PS1 and also end at PS1. Final electron acceptor for non-cyclic is NADP+, and cyclic is PS1. The product for non-cyclic are NADPH and ATP, but for cyclic it is only ATP.
Linear electron flow. First, a photon, which is energy from light, hits a pigment and its energy is passes among pigment molecule until it excites to P680. Second, an excited electron from P680 is transferred to the primary electron acceptor, which is P680 is missing an electron and become P680+, which is very strong oxidizing agent. Third, H2O is split by enzyme and the electrons are transferred from the hydrogen atom to P680+. Thus, it reduces to P680, which is P680 plus accept an electron and become P680. Then, O2 is released as a byproduct of this reaction.
Each electron falls down an electron transport chain from the primary electron acceptor of PS2 to plastoquinone, from plastoquinone to cytochrome complex, from cytochrome complex to plastocyanine, and from plastocyanine to PS1. During the passage of electron from plastoquinone to cytochrome complex, cause the pumping of H+ from stroma to thylakoid space. This creates proton gradient across the thylakoid membrane. Diffusion of H+ across the membrane via ATP synthase which is from thylakoid space back to stroma, through ATP synthesis. The energy released is used to make ATP from ADP and inorganic phosphate.
Hi! In PS1, the light transferred from the PS2 excites the P700, which this is because the P700 before that loses an electron to the electron acceptor. Then the P700 is being reduced to P700 plus, which is then accepts an electron passed down from PS2 via the electron transport change. Each electron falls down an electron transport change from the primary electron acceptor of PS1 to protein pyridoxine. The electrons are transferred to NADP plus and reduced it to NADPH. This process removes H from the stroma. The electrons of NADPH are available for the creation of the next Kelvin cycle. The electrons are then transferred to NADP plus with the aid of NADP plus reductase enzyme to reduce it to NADPH. Then, this process removes H plus ions from the stroma, causing the electrons of NADPH are then can be usable, reusable, and available for the next reaction of Kelvin.